I think it's upside down.
How is it possible to
Let's see what we can do. Just bend that up just a little bit for you. Feel okay?
Good, very good. Thank you.
Thank you.
Are we on? Thank you. I think, there we go. Now we're on. We're running just a tad bit late, and so I will go right to our panel discussion and make my announcements at the end.
Yeah.
Great.
But that doesn't take away from you.
Welcome back to everyone. I wonder if any of the panelists would like to ask Dr. Bertozzi any questions or offer any observations. Go ahead.
Karen, and thank you very much for telling this such a beautiful and fascinating story. I almost want to eat an M&M right now. I've two quick questions. One is that so it seems to me the glycans and the cell membranes of cancer serves as almost like a barcode for these cancer cells. My question is whether different type of cancers, different tissues have different type of glycans. My second question is whether other components on the cell membranes, like there are many lipid species on the cell membranes, there are also proteins. Can they also serve as a barcode, like a glycans? How they're interacting with each other?
Yeah, thank you. Excellent questions. So to your first question, there's a handful of stereotypical changes in cell surface glycans that have been associated with cancer. The sialic acid phenotype is probably the most prevalent across different tumor types, and now we have a theory, as to why it arises. With the other phenotypes, we don't know anything about their functional significance. They're just correlations with malignancies, and they tend to be less prevalent, more specific. For example, there's one in particular in colorectal cancers, but you don't see it in other solid tumors. And nobody has any idea so far what the biological function is of those. So there's plenty of mysteries to solve in this field. And hopefully, not just my lab, hopefully many labs are mobilizing around this. And yes, there are proteins, for sure, that are associated as cancer markers. In fact, most of our tumor-targeting antibodies are against proteins that are either exclusively or preferentially expressed on cancer cells. And there are interactions between them as well. So there are glycoproteins. In fact, most of the proteins on the surface of a cell are carrying glycans. They have glycans attached to them. And so there might be settings in which it's not just the glycan or just the protein, but the combination of the two that gives specificity to cancer. And at lunch, I was sitting next to some of our audience members who came down from the Hormel Cancer Institute, who are working on a particular glycoprotein called MUC1. Doesn't matter what that means, but what makes that interesting in the setting of cancer is the combination of protein and sugar. Yeah, it's pretty interesting.
Other questions from the panelists? If not, maybe a follow-up question from the audience, which is, are there other non-cancer related diseases for which study of glycans are being explored?
Yes. Cell surface glycans are important in pretty much all areas of biology. They contribute something. Where they're quite famously important in diseases is in virology, There are viral pathogens, viruses, that attach to those sugars as a first point of contact, and that's how they enter your cells. And influenza, the flu, is a classic example of that. And by the way, guess what the flu binds to? Sialic acid. And the flu drugs that are on the market, I don't know if anyone here has taken Tamiflu. Anybody taken Tamiflu? That is a sialic acid analog. It looks very much like sialic acid if you drew out the chemical structure. And it interferes with the interaction between the virus and those sialic acids. But the other is chronic inflammation. So I rattled off a bunch of inflammatory diseases right then. And in those diseases, immune cells are able to infiltrate the tissue and cause the damage, but the way that they infiltrate has to do with binding to some sugars in the blood vessels around the inflamed tissue. Those are the sort of famous examples where the pharmaceutical industry has put a lot of work into making drugs and so on.
A question from the audience also. Is there any connection, or is there any connection known, between dietary intake of the sugars that you talked about at the beginning and the sugars that coat our cells? Are there any things that we can even talk about?
You know, the smart money says yes, because in biology, everything is a system, a connected system at some level. Sure. And it's true that you can shift the pattern of cell surface glycans by changing the supply of simple sugar building blocks in the cell media. That's for sure the case. The question as to whether that's physiologically relevant in the human body, I can't answer that. I don't really know. But you want to, Frank, maybe you can contribute.
Well, so this is something I'm not sure whether it's directly related to glycans or not. We know that advanced glycation products is a major risk factor for diabetes and diabetes complications. We have measured blood levels of advanced glycation products or AGs, and those are very powerful, potent risk factors for diabetes and cardiovascular disease. And we also know diet has a huge influence on the circulating of AGs. So people who consume this Western, typical Western diet, high amount of sugar, especially sodas, and also even diet drinks, they have elevated levels of AGs. And also we know that our tissues, especially heart and the kidneys, have AGs receptors. So the AGs in the blood bind to those receptors, which cause subsequent complications in terms of cardiovascular complications and renal complications. So not exactly sure about the molecular mechanism in terms of how the dietary sugars bind to the proteins and how that create some kind of, you said earlier, this interaction between our proteins and the sugars in the body.
Yeah. And by the way, if you Google, or ChatGPT or whatever you all do, if you Google like sugar and cancer, you'll get like a huge bunch of hits, both scientific literature and pop culture literature, because there's this thing called the Warburg effect, if you've heard of that. And it was discovered by a famous German cancer biologist named Otto Warburg, who was kind of active in the 1930s, 1940s. And Warburg discovered that cancers, as they advance, can become addicted to sugar, where they need to get their energy to divide and proliferate from sugar. They're not able to get that energy from other dietary sources. And based on that, it's been suggested that if a cancer patient went on a very, very low carb, low sugar diet, this might slow the progression of their cancer. But I don't know if anyone has actually done a legitimate clinical study.
Yeah, ketogenic diet has been used in cancer treatment. It has been shown that ketogenic diet can enhance the PD-1, the immune therapy for certain cancers, but those trials are relatively small.
Yeah.
And I think it's pretty clear that the favorite fuel for the cancer cells is glucose.
Yeah.
And the question is, how can we eat a diet that can help keep the body healthy, but also reduce the fuel for cancer. So ketogenic diet may be one of the potential strategies.
Professor Runge?
So in your introductory description of a normal cell and a cancer cell, your metaphorical description of the difference implied a kind of movement from order to disorder. Is that intentional in the sense that the increased proliferation of the sialic cells in the cancer case is a reflection of some type of degradation, entropic or otherwise, or alternatively, is the cancer cell actually mounting an organized defense?
Yeah, it's a good question. We have looked a little bit at, something must have changed inside the cell to lead to the changing of the cell surface garden, right? And what is that? And we have looked at some common oncogenes. So there are certain problems inside the cell that are common for lots of cancers, and one of them is that cancers can overexpress a transcription factor that drives the expression of genes that cause the cell to become cancerous. One of those is called MYC, M-Y-C, MYC. And some 50% of human cancers have overproduction of this MYC transcription factor, and because of that, there's genes that are getting expressed at higher levels than they should normally be expressed. And it turns out in some of these MYC-driven cancers, one of the genes that's getting overexpressed is an enzyme that puts on sialic acids onto these glycans. So we know for MYC-driven cancers, there's an upregulation of a transcript, which increases the flux in a biosynthetic pathway, which leads to the sialic acid phenotype. That's one example where we've mapped what happened inside the cell that led to the change on the cell surface. But there are many other cell types where MYC is not a factor, and we don't really know what happened in there. But yeah, it might just be part of a program which comes ultimately from an oncogene.
Here's an inevitable question that, okay, maybe I'm interested in it. If we did taste glycans, would they be sweet?
As chemists, we're always taught, don't eat your molecules. Generally a good rule of thumb. Having said that, we're not on Zoom or anything. I'll just say, back in the 1980s, when things were a little looser, me and my lab mates in graduate school, we did actually taste we made sugars, we synthesized chemical analogs of sugars, and we did taste them, actually. Only if we could recrystallize them from high-grade solvents that were like ethanol, things that were friendly, and so on.
Of course.
We didn't drink the ethanol, just FYI. But we did, and I have to say, glucose is not that sweet compared to some others. The sweetest that I remember from tasting those things was galactose. Galactose is sweet. Glucose is just kind of there, in between. And then some of them are bitter, like mannose is bitter. Sialic acid is bitter. So,
Interesting. Thank you. Thank you. Okay, I'll admit that was my question.
Don't do that. So.
Don't try this at home, please. Another question that maybe is more of an all play, and that is, how have changes in how the CDC, the National Institutes of Health, and other federal grant makers have been making awards had an impact on your research agenda or your current ability to fund your research?
Oh, how much time do we have? So up until now, I've been working in this area of science now for almost 30 years, and the reason this was possible is because I had pretty sustained, uninterrupted research funds from the National Institutes of Health. And 2 of my NIH grants are from the National Cancer Institute, which is one of the subdivisions in the National Institutes of Health. This year, everything has been kind of turned upside down, where I don't think many of us have a view as to whether that kind of sustained funding can be counted on looking forward. Maybe it can, maybe it can't, we just don't know. For example, the White House issued an initial budget proposal that would have cut the NIH budget by 40%. And so you can do the math, right? If that means my lab budget would also have been cut by 40%, I would be talking about something else. I wouldn't have been able to do this research, probably. It would be even worse if my lab were funded by the National Science Foundation, the NSF, because the proposal on the table is to cut that budget by 55%, but not evenly across all scientific disciplines. The proposal is cut the NSF by 55%, but cut chemistry or chemical sciences within the NSF by 75%, so that other areas can be maintained at 100%, such as AI, quantum computing, right, things of priority to this administration. So if that budget had passed as proposed, then we'd be in dire straits right now. The reason we're not, basically ready to just give up, instead we're sort of uncertain, is because there was a counterproposal put forth by the House and the Senate, which proposed to restore those two budgets to their current state. And we don't know where this is going to land, and now the government is shut down, right? So I think right now, the best, I guess I don't know how you guys feel, but I feel like uncertainty is the prevailing feeling, just uncertainty. But when you're uncertain, you can't make commitments, which means you can't hire students and postdocs. You can't, certainly graduate students that need a 5-year runway, you don't feel confident that 5 years from now you'll still be able to pay their salary. So that's the problem right now. So, if you haven't already, please reach out to your senator and your congressperson. This outreach matters. Feel free to call their office, or send them an email.
Did either of you want to comment on that at all from your vantage point?
I mean, you're in the belly of the beast right now at Harvard.
I don't want to talk about it.
I don't blame you.
Well, Harvard grants have been terminated, and it's unprecedented. I think it's real. It's already doing, real damages to science and technology, our global leadership in science and biomedical research and educating the next generation of scientists. But I think we have to play a long game, and we have to take a long view. And my hope is that we will get out of this. We don't know how long it will take, but I think our country is resilient, and the science is resilient, and eventually I think the system, hopefully the voters will be able to put science back in track. But it is currently a very challenging time, and we are working very hard to make sure that our core research programs are still operating, and make sure that our PhD students are still being supported, and make sure that the faculty that we have recruited in recent years will not simply leave. I know many other countries are recruiting our talents away, but we were doing everything we can to retain those talents.
A slightly different question. I'm aware of the fact that we won't have Dr. Bertozzi with us tomorrow, and we have spent a day thinking about these questions from the life sciences, and so I'm wondering if all three of the life scientists might have anything to say about the question of how, well, it was put here as, what's the trick to making this accessible to a wider audience? I think all of us would agree that the ideas presented to us today were presented in a way that made us feel really smart because we understood them, right? I mean, it's an amazing accomplishment and a real challenge. And when I look back at the history of the Nobel Conference from 1978, when I started going, let me tell you, you understand what it means to speak to a general audience. So can any of you speak from your vantage point about why that's important, how you've come to understand that, and maybe invite our, give us a little bit of a peek behind the curtain about how you've come to understand how to do that work, which all of you did with aplomb today.
Well, I could start by just saying I think that as a scientist, at least back when I was being trained, we were kind of trained to just be quiet and stay in the lab, do the work. And it's really only, I would say, in recent years where we're seeing more and more scientists kind of getting out there and talking about the work. A lot of the science communication had been traditionally left to science writers or science reporters, which is still a wonderful thing. But I think when you can get out there and talk about your research and get people excited about it, especially students and younger people, and explain it in a way where they want to learn more and they want to get on board with it, I think that that's a critical element now to our jobs as scientists and educators. So I encourage all of my students to, be able to talk about their research, not just to the scientists and to the, professors in the room, but to their roommate, to, the friend on the team, and really just to be able to communicate this in a way that makes people get excited about it, because that's going to keep it going forward. Otherwise, we're really in a vacuum, and that's not a good place to be.
Anything else from you? Please.
I mean, as you all know, nutrition is always a very popular topic. It's a hot topic, and it's on the news all the time. And currently, as you know, because of the social media, there's just so much misinformation, and I think it's not just misinformation and a lot of confusion about nutrition. So that's why nutrition communication has always been a top priority of my department. And the Nutrition Source website that I mentioned earlier, I think, has played a very important role in terms of disseminating evidence-based nutrition information to the general public, policymakers, and the health professionals. But that's not enough. I mean, we cannot really compete with the social media influencers. And so currently we are working with a media communication team at the school to create more AI-friendly website, because nowadays AI just grab information from various websites and give the information to people who are seeking them. So how to make the website, the information can be more usable for people who are using AI to get this kind of information. And also, I think it's very important to communicate our scientific findings to policymakers, because at the end of the day, I mean, on a lot of nutrition, food-related topics, we do need evidence-based policies, evidence-based recommendations. In 2015, I had the privilege to serve on the USDA Dietary Guidelines Advisory Committee. And the dietary guidelines for Americans, as you may know, are updated every 5 years. And the scientific report put together by the Dietary Guidelines Advisory Committee serve as a scientific basis for the dietary guidelines for Americans. And it's a lot of work. I mean, basically 20 leading experts from different fields spent 2 years create this report. And then the government translate that report into the dietary guidelines for Americans, which are very important because the SNAP program, the WIC program, and the school lunch program, they all have to follow the dietary guidelines. So we spend a lot of time make sure that the scientific evidence is not just included in the report, but also is more digestible and more can be well received by policymakers who don't necessarily have nutrition background. So this kind of science to policy, of course, is not a straight line. I mean, there are a lot of back and forth and a lot of curve, linear pathways. But the evidence is always the foundation. So we want to make sure that we create the best available evidence, make sure that this kind of evidence is available to policymakers, make sure this evidence is also accessible to the general public. But again, we still have a long way to go in terms of how to bridge the huge gap between the science and policies.
Mm-hmm. Dr. Bertozzi, do you have any thoughts about that? I've told it's not an M&M, it's a forest analogy many times. It's memorable. So do you have any thoughts about how you've come to be a communicator?
I think, well, I love a good story. And I try to tell a story, right? And so for me, I think it's just, I don't know that I was trained to communicate. You go to the lab and do your work. But I really have come to appreciate when I'm trying to learn about a field that's outside of my kind of comfort zone. I really appreciate it when there's a good story there, because I can remember it. It sticks better. So my goal, if I'm talking about something very technical, is to figure out how do I turn this into a story. And a story has a beginning where you introduce the characters. There's a middle where the tension builds up. And then there's an end where it's resolution and yay, happy ending, hopefully. Right? So,
That's great.
If that was effective, then thank you for the positive feedback.
It worked for sure. This is a wonderful illustration of the incredibly complicated ways that science and its social and ethical implications are around us all the time, right? I mean, just think of this conversation this afternoon. I'm going to turn it over to Phil.
That's the perfect time to. It brings us to our natural stopping point, and I almost can't resist to say one other way is to attend a liberal arts college. In my own department, I can't resist a shameless plug teacher called Communicating Science. So that's another way that we can help this out. A couple of quick announcements before we leave. As you exit, you may have noticed on the lawn there is a live dance performance, Sugar, Because I'm Lonely. And the dancers are Kaya Meyer, Marissa Schmidt, and Johanna Mai, who's one of the liaisons. It was choreographed by Jill Patterson, and the cellist is Annalise Myers. So catch that on your way out. Also, immediately following this, down in the outside the bookmark in the basement of the Jackson Campus Center, Tom Annesley will be signing copies of his book, Beer and the Nobel Prize, Tales of Nobel Prizes and the People Who Won Them. So you will have a chance to get an author signing from that. This evening from 5 to 7, there's a reception for an art show called Strangeness Should Flow Like Sugar. This is in the Schaefer Fine Arts Gallery on the south side of our campus for visitors. That's that way. Just walk until you see pretty much the last academic building. That will be the Schaefer Gallery. And it features at 6 p.m. a talk by artist Anda Tanaka, who uses a sugar relief process to make prints. So I don't know much more about that. And then if you're still around the campus at 7:30, The Moth in the Bjorling Recital Hall, which is also on the south side of campus, they are doing a slam, and the topic is cravings, which again, I've been focused on that donut all day long. So I hope to see many of you there. We will resume again tomorrow. Doors will open at 8:30, and the talks will start at 9. Thank you so very much.
Hey, everybody. Wow, what a day. One more time, I'm Tane Danger.
I'm Maggie Kurth.
We are wrapping up day one of the 61st Annual Nobel Conference at Gustavus Adolphus College. Just had our final panel conversation. I should say day one, because we're going to be back tomorrow. But Maggie Kurth, final kind of things from today that are just really sticking with you.
Man, sugar is important.
Yeah.
Sugar is so important to your body in ways that I never would have guessed. And I think it was really great to wrap this up with that presentation about how sugar functions and a shell around your cells